Hi everyone, welcome to today's class. Again, my name is Andrea Joss and I'm teaching you the protein section in your 40/20 class. Just to recap a bit, this is class 3 out of 5 about protein, and last class we talked a bit about what proteins were, what amino acids are, and protein structures and denaturation and mutations and things like that. And so today in class 3 we're going to spend time talking about what happens when you actually eat proteins, so the digestion and absorption of proteins, and then how we can measure the amount of protein that our body is using at a particular time or over the course of a day, let's say, to assess whether we are eating enough protein. And so at the end of the day, you'll see how this recommended dietary allowance, or the RDA that I talked about in the first class, is actually derived by these nitrogen balance studies that I'll talk about. Okay, so let's talk about digestion and absorption of protein. So these are some key points. You'll see the next couple slides. They kind of look like this, and they go through step by step these points of digestion and absorption. So we'll start with digestion of protein. So when you eat protein, obviously you chew, that starts to break things down a little bit, the mastication, and then you swallow and it goes into our stomach. And actually protein is the macronutrient that is digested in the stomach. None of the other macronutrients are. And this is because the stomach is acidic, and I'll explain to you why that's the case. But carbohydrates and fats are not digested in an acidic medium, only protein is. So protein digestion does start in the stomach, and like most things, like most procedures in the body, they are under hormonal control. So the hormone gastrin is the first hormone that really gets things going, and what it does is, when it senses that there's some food in your stomach, it triggers a whole bunch of different things to occur. So one of the first things that it does is it gets the stomach to start churning, and then it gets gastric juice to be released. Now the interesting thing about gastrin is that you can actually trigger the hormone gastrin just by looking at food, or by even smelling food, thinking about food potentially, particularly if you're hungry. And so if that happens, you can actually start this process of churning of your stomach and releasing of gastric juices when there's no food actually there. So if your stomach is rumbling, like if you're hungry, it may be because you just thought about food and your hormone gastrin is actually starting to get the stomach ready to eat. Or if you go to a restaurant, which unfortunately a lot of us have not been to the past little while, and you see and smell food, you see people eating food, it gets you hungry and it gets your stomach going. That's the hormone gastrin. Okay, so this is what it does. As I said, it triggers the churning of the stomach and it starts the breakdown of the food inside the stomach. It also has a very important job of releasing gastric juice. So this is a amalgamation fluid, if you will, of various different things that come together within the stomach. So one of the main components of gastric juice is HCl, or hydrochloric acid. And you can see here HCl is secreted by the parietal cells within the stomach. Along with HCl secretions, we also have mucus that's secreted within the stomach, and the mucus is secreted by the goblet cells. And the mucus has a very important role in the stomach, which I'll get to in a moment. And in terms of breaking down protein, you obviously need the enzymes that act on the peptide bonds and other such bonds that have occurred within the protein to make it its structure. So these particular enzymes are also part of gastric juice, and they're released into the stomach. And the one that acts the most in the stomach, the primary one that breaks down protein, so the initial protein breakdown, happens with an enzyme called pepsin. So you can see that down here. But pepsinogen is actually what's released from the chief cells into the stomach. So I'll explain this in a moment. What are the roles of these these particular parts of the gastric juice? Well, HCl, or hydrochloric acid, acts to denature the protein, and it also acts to convert pepsinogen into pepsin. So remember we talked about protein denaturation. It can happen at various different pHs, and what it does is it kind of unravels the protein. So this is really important. This is a really important aspect of protein digestion, because if the protein is like this and the amino acids are really tight together, then the enzymes are not able to get inside all the little crevices to break down the amino acids appropriately, and therefore we can't absorb the protein at all, or the amino acids. So HCl is a very strong acid, and it's very important that the proteins become denatured and unraveled so that pepsin, the other enzyme, can actually get in there and start breaking down the protein into smaller peptides. So you can see here that this is what happens in the stomach. It's broken down into smaller peptides in an acidic medium. Now, I said I'd get back to this, but why is mucus important in the stomach? Well, remember, the stomach is also an organ. It is made from protein as well. And so if there's no mucus, or there's no barrier, there's no lining of the stomach in which the mucus actually provides the barrier, the enzyme pepsin is going to actually break down your organs. It's going to break down your stomach. And this is sometimes what happens when people have ulcers. The mucous membrane is disrupted, and the stomach enzymes and hydrochloric acid actually break down parts of the stomach for various reasons. It can also happen because of H. pylori infections and other such things, but we're not going to get to that right now. But the point is that the mucous membrane in the stomach acts as a barrier to the stomach from being exposed to hydrochloric acid and protein digestive enzymes. Here's another question that I have for you that I want you to think about for a moment. So why do the chief cells of the stomach secrete pepsinogen? If you notice back here, I said the enzyme that's secreted by the chief cells is pepsinogen, and HCl converts pepsinogen into pepsin. So why would this be the case? Sorry, my nose is quite itchy. I'll give you a second to think about it. You can pause me and think a little bit more if you'd like. Anyway, I'll tell you the reason why. Well, pepsinogen is actually the proenzyme, so it's the precursor enzyme to pepsin. And so in the chief cells of the stomach, it has to be stored as the proenzyme, which is the inactive form of the enzyme. Because what would happen if your stomach stored active enzyme that breaks down protein? It would break down your stomach on a regular basis. So that is why pepsinogen is actually secreted from the chief cells, and it's turned on, or switched on, if you will, by the hydrochloric acid within the stomach only. Okay, so that is the reason why we secrete pepsinogen, which is then turned on, or activated rather, by hydrochloric acid to become pepsin. Okay, so continuing on then, these partially digested proteins, so these smaller peptides, and of course other food stuff that hasn't been digested, but it's been churned a lot. So we're talking, anything else that you've consumed with the protein that's in the stomach at the same time. It actually forms this sludgy compound called chyme. And chyme is acidic, obviously, because of the hydrochloric acid that was added to it. But the chyme then enters the small intestine through the pyloric sphincter. And at this point, gastrin levels are going down, so the stomach is stopping to churn, and the gastric juice is not being released as readily because it's already done its job. There are other hormones that take over at this time. So once the chyme, or the acidic chyme, enters the small intestine, it needs to be neutralized right away, because no other enzymes can work in a acidic medium, and you don't want this hydrochloric acid in and around all of your other organs. So secretin or secretin is another hormone that now acts at this point. And the main thing that it does is it stimulates the pancreas, so one of these accessory digestive organs, to release bicarbonate into the small intestine. And so bicarbonate is released into the small intestine, and this acts to neutralize the chyme so that digestion and absorption can occur within the small intestine. And another hormone is CCK, or cholecystokinin. And CCK stimulates the pancreas as well to release other enzymes. And these are the other enzymes that break the protein down even further. Okay, so the pancreas releases its own type of juice, if you will, into the small intestine. And this time it's not pepsin, but it's trypsin and chymotrypsin. And these particular enzymes, they also digest peptides into smaller units. So if there's polypeptides, oligopeptides, dipeptides, tripeptides, anything like that that is still within the chyme, these other enzymes now act upon it within the small intestine. And trypsin and chymotrypsin need to be, they function in a neutral environment where, remember, as I said before, pepsin is in a acidic environment in the stomach. All right, so moving on then, now that we have these peptide bonds being broken down even further, they're broken into smaller units, so either, dipeptides or tripeptides at this point. There are other brush border enzymes that further hydrolyze the proteins into individual amino acids. So these particular enzymes are at the luminal border of the small intestine. So this luminal border is characterized by villi, and I have a picture in a subsequent slide about what a villi looks like, or what the luminal border actually looks like. But this is the site of absorption of the amino acids. Okay, so now that digestion is completed, the last two enzymes that further break down the dipeptides into individual amino acids are carboxypeptidases, which cut at the carboxylic acid end, and the aminopeptidases, which can cut at the amino end. But both of them work to create the individual amino acids, which are then absorbed into the enterocyte of the small intestine. Okay, so most of the amino acids at the brush border are absorbed in the proximal 1/3 of the small intestine. So this is the duodenum and the upper portion of the jejunum. Okay, the small intestine is really, really long, but actually most of the absorption of macronutrients, and also, as I'm saying here, amino acids, are done in the proximal third. Okay, enterocytes, so the small intestine cells, actually have specific amino acid transporters, and this is how the amino acids get into the enterocyte. They also need to get from the enterocyte into the bloodstream, which we'll talk about in a moment. But when they go from the lumen of the small intestine into the enterocyte, they use amino acid transporters. And most of these amino acid transporters work via active transport. So they use a sodium-dependent mechanism of some sort, a sodium-potassium pump. So it does require energy to transport these amino acids into the enterocyte. And something interesting to note, so several of these amino acids compete for the same transporter, or they have the same transporter. And on a normal basis, when you're consuming normal amounts of protein, this is not really an issue. You'll end up absorbing over time a fair amount of most of the protein that you do consume, which is great. But this becomes a problem sometimes when you ingest large amounts of one particular amino acid or one group of amino acids, like branch chain amino acids, for example. This is all the rage in the gym these days, or it has been for quite a while, and I put dietary supplement use just as an example of this. People know, and research shows, that the branch chain amino acids, particularly leucine, is really important amino acids for the stimulation of muscle protein synthesis. And so people experiment with actually taking leucine, crystalline amino acid, or branch chain amino acid supplements, which is leucine, isoleucine, and valine. Okay, and so when you do that and you kind of flood the system with one particular amino acid or one group of amino acids, this is when you tend to see issues with absorption of other amino acids, particularly those that share transporters. So if you're consuming too much leucine, you're going to not absorb the isoleucine and valine. And that's a problem because all three of them are essential amino acids that our body does not make. And so you need, like, even though leucine may be the king, if you will, or the number one amino acid that switches on muscle protein synthesis, all of them are needed to produce new proteins. So, it's one of those things to think about when you're going to supplement with a single amino acid, the ramifications of that with respect to, actually having it do what you want it to do, which is improve protein synthesis. You need all the proteins to do that. So just something to keep in mind. All right, so moving on then, it's important to also bring this part up. So sometimes there are some intact small peptides that do get absorbed into the enterocyte, so through the villi. So it's not always amino acids. Sometimes small peptides like tripeptides or dipeptides do get through. And they can be broken down further in the cytosol of the enterocyte through these brush border enzymes, but sometimes they don't. And they can then be secreted into the bloodstream and they make our way into the systemic circulation. That can happen. And in some instances, though, these small peptides can cause illness or allergy. So when our body mounts an allergic reaction, it's usually to a dipeptide or tripeptide from a particular food that has gotten through the digestive system. And there are some allergenic foods, of course, as you know, that we need to be aware of. Nuts and shellfish and eggs and dairy, wheat gluten, things like that. These are all protein sources that can mount allergic reactions, and it's because there are small peptides that have gone through and caused the allergic reaction. There's also illnesses that are hallmark of these issues with the tiny peptides going through the digestive system and ending up in the bloodstream. Irritable bowel syndrome, inflammatory bowel disease and Crohn's. Celiac disease is more of the allergic reaction actually because it's an allergy to wheat gluten. But inflammation can also be caused by these little peptides that get through. And so just to put that out there, that can be a source of illness, inflammation, and allergic reactions. Okay, so continuing on, now we'll talk about the absorption and fate of amino acids. So now that they are broken down into the amino acids, they've actually been absorbed by the small intestine. As we can see, oops, sorry about that, as we can see here. They then go into the capillaries within the villi of the enterocyte. And the capillaries, all of them within the villi, which I'll show you in the next slide, I believe, all of these converge into venules, and then they dump all of their contents into the hepatic portal vein. And the hepatic portal vein drains everything from the small intestine and it takes it right up to the liver. This is an important point because the liver is our detoxifying organ. It is our organ that sees everything essentially, actually besides some large fat molecules like the larger fatty acids, but you'll learn about that later because those travel through the lymphatic system. But in any case, almost every other type of food that we eat or a lot of the vitamins and things, those go through the liver first. Of course, as I said, there are those that don't, but again, you will learn about this from Dr. Cedia in the class. So once they get to the liver via the hepatic portal vein, they're used for various different functions. So they're used for energy production and oxidation. But remember I mentioned before, there are many other uses for protein. The use of protein for energy is lower on the list in terms of priority, but it does happen. Proteins can be used to synthesize glucose, and this process is called gluconeogenesis. And so there are particular amino acids that are gluconeogenic, or that can be used to make glucose. You can also synthesize non-protein nitrogen-containing molecules like neurotransmitters and other types of enzymes and other such things, which is one of their main uses, is to synthesize new proteins. So synthesize new body proteins. Protein turnover is constantly happening in our body, and actually the turnover rate is quite high for many of our organs. We also know that protein turnover happens in the muscle, and so there's amino acids that are used for that as well. Our gut has a really high rate of protein turnover, so it's constantly being broken down and remade, and so amino acids are used for that as well. As I've written here, most of the repackaging and formation into new molecules happens in the liver, so a lot of the making of the particular enzymes that our body requires throughout all of those types of things do happen are made in the liver. I talked about lipoproteins in the last class, and these are the carrying molecules for fatty acids and triglycerides that you'll learn about when you do the fat section or the lipid section. But proteins are also a big part of those molecules too. So they're all packaged in the liver, except for chylomicrons, but that's a whole other story. In any case, I also made the point here that branched chain amino acids are spared by this repackaging for the most part by the liver, and they are generally pumped out of the liver and they make their way to skeletal muscle. So branched chain amino acids are used preferentially elsewhere in the body. And this is part of the reason why leucine is such a strong trigger for muscle protein synthesis. Remember, leucine is one of those branched chain amino acids, and it is preferentially used by the skeletal muscle. So as I've written here, they are spared by the liver, they're put in the bloodstream, and they get to go to the muscle to be used for protein synthesis, or to turn on the machinery for protein synthesis. Okay, so here's some visuals for you with respect to the digestive system. So here is the inside, actually, I'll start up over here. This is a picture we can see here of the hepatic portal vein. So this is that vein that collects all of the absorbed nutrients from the small intestine. Okay, so we can see it's converging over here from the venules in the villi, and then it converges into this hepatic portal vein and goes up to the liver. Okay, I actually should have started down here now that I'm thinking about that. But in any case, here's a picture of the small intestine. You can see it has muscular layers in it to help push the food along as it's being absorbed. There are various processes called peristalsis and segmentation, if you've heard about that. It kind of looks like a worm is being, like, if you've ever watched a worm move, it's kind of looks like what happens in the small intestine. There's areas that bunch and then release, and then bunch and then release, and it helps to kind of churn things inside to allow for maximal rates of absorption. Also, we can see here there's these circular folds within the small intestine, and this also increases surface area for absorption. And so this is a blown up picture of the lumen, or the brush border of the small intestine, and these are the villi. So each one of these is a villi, and each one of these small things in here are microvilli. these small cells, and these are the enterocytes. All right, and inside each of the villi is a network of capillaries and veins, and this is the site of nutrient absorption. So this is where it occurs. So nutrients are absorbed actually in two places. They're absorbed from the lumen of the small intestine into the enterocyte, and then they're absorbed from the enterocyte into the bloodstream within the villi. And at both occasions, there are amino acid transporters that do this. And then from here, going back to this figure then, you can see here that the network of blood vessels converge into this hepatic portal vein that goes up to the liver, and then the liver has its first pass at everything that comes to it, and then is able to put the rest out. Okay, so protein synthesis. I'm almost done actually for today's lecture, but well actually no, there's one other topic that I wanted to talk about today. But so now that we've absorbed our amino acids, now that they're in the bloodstream, they need to go, or in the liver rather, they need to go to where they need to be in order to produce particular proteins. And this is like the central dogma of biology here. This is a protein synthesis. We go from a gene so a gene is transcribed, and then it's translated, and then you make a protein. So DNA, RNA, protein is the central dogma in biology, I would say. And so, I don't want to get into this in too much detail because you probably have already when you've actually taken your biology course, which I assume most of you have already. But in any case, this still holds true for all of the proteins that we make in our body. So our DNA has the genetic code for making our protein. The protein is transcribed into messenger RNA. The mRNA takes the code from the nucleus to the ribosomes, which are contained within the cytosol. And the transfer RNA translates this genetic code from the mRNA and brings the amino acids to the ribosome, and the ribosome makes the polypeptide chain. Then this chain then undergoes a series of modifications, as we already talked about, and turns it into its tertiary and quaternary structure, and then you have your protein, which then goes and does what it needs to do. It's pretty simplified, but I'm pretty sure you've gone through this in more detail. I wanted to also mention that, we are a kinesiology program, so I wanted to mention specifically one of the fates of our amino acids is, sorry about that. One of the fates of our amino acids are to make muscle. And so we have here our skeletal muscle mass over here. So amino acids, as I said, are used to make muscle, but I think it's also important for us to understand that while these amino acids are building blocks for muscle, there are several other factors that are involved in skeletal muscle growth. So it's not just when amino acids are there, you end up making muscle, but a lot of these processes, like I've mentioned before, are under control of hormones. So we have anabolic hormones and we have catabolic hormones that in the presence of amino acids can act in various ways. So with respect to the anabolic hormones, we have three main ones: insulin, growth hormone, and testosterone. And these three hormones are anabolic, as I said, so meaning that they build tissue and they store nutrients. So in the context of muscle mass, insulin acts to help to build muscle. Actually, the main role of insulin with respect to skeletal muscle hypertrophy is more in preventing muscle breakdown, but growth hormone and testosterone both act to increase hypertrophy. With respect to catabolic hormones, the main one is cortisol. And so this is why in situations of stress, various different situations, including illness and injury and starvation, and just stress in general, we have a cortisol release. And this actually is catabolic. So it acts to break down our muscle to provide amino acids to the system to still aid in to still aid in protein synthesis in other places of the body where it's needed. So that's kind of what cortisol does. Whereas insulin, growth hormone, and testosterone are the anabolic hormones, which act to build muscle in various ways. Cortisol is a hormone that acts to atrophy muscle. So you don't really want high levels of cortisol. So then you can ask yourself the question, now that we know, how how protein is digested and absorbed, getting back to what I talked about in the first class, we can ask ourselves this question, are we getting enough protein? So are we providing our body with enough protein in order to sustain all of the protein synthesis that it needs to do to provide us, or to provide our body with all of the protein-related molecules and functions that it needs? We have an RDA, so we have a recommended dietary allowance. And the idea, remember as I mentioned before, is that if we eat at the level of the RDA, then this is enough to, this is supposedly enough for our body at a level that's going to prevent disease. So remember, I talked about this in the first class. And the RDA was actually derived from nitrogen balance studies, which I'll get to on the next slide. But just to introduce this idea of nitrogen balance first. So we can use our nitrogen balance in the body. We can measure nitrogen balance to assess how proteins are being used. So as I've written here, it's a measure of the relationship between the amount of nitrogen we take into the body, and this is mostly as food, and the primary nutrient that contains nitrogen is protein. So it's essentially a measure of the amount of protein that our bodies are taking in. And then you measure the amount of nitrogen that is leaving our body. So, nitrogen can be lost in various different ways in the body. We can lose it as urine, we can lose it in feces, we can lose it from our skin, so like skin cells being sloughed off. If we lose hair, I have curly hair and I lose a fair bit of it. I just pulled out a piece of hair here, but for the most part, like that is a source of nitrogen loss as well. Nails too, if you grow your nails and then you cut them, that's nitrogen being lost because it's a type of protein. So there's many different ways in which nitrogen can be lost in the body. And if you put those two measures together, nitrogen in and nitrogen out, then you get a measure of whether you are consuming enough protein. Okay, so here's some context over here. So when we do nitrogen balance studies, we're assessing whether the amount of protein in the body is constant, decreasing, or increasing. So if your intake of nitrogen is equal to the excretion, then your body is in nitrogen balance. It means that you're providing your body with the right amount of protein for the particular situation. If your intake is less than excretion, then you're in negative nitrogen balance. This means that you're excreting more nitrogen than you're taking in. So you're breaking down body proteins to be able to excrete it. Okay? If you are in positive nitrogen balance, it means you're taking in more protein than you're excreting. So you're actually retaining the protein in your body more. And so there's various different situations in which one would be in negative nitrogen balance versus positive nitrogen balance. So let's think about these for a second. When would you be in a situation of negative nitrogen balance? Okay, so there's a picture on the slide there that kind of gives you a clue. This one over here, this is someone probably in negative nitrogen balance. And generally, we lose body protein, or we lose nitrogen more than we're taking in if we're stressed, so we have that cortisol response. If we have an illness, if we have an injury, if we have a burn, if we have an infection, if we're immobilized, those types of situations. For a couple reasons, our bodies are working overtime, but also with illness and with a bunch of these other factors that I mentioned, we're generally not eating well either, so we're not taking in enough protein. And in order for our body to heal itself properly, it needs more protein. So the point is that if we, with respect to nitrogen balance, if we're excreting more than we're taking in, then we're in negative balance. Now, with respect to positive nitrogen balance, this is a figure that I have on the slide. So a woman that is pregnant is in positive nitrogen balance because she is growing a fetus inside of her, and that fetus is taking protein. Okay, so other ways in which one would be in positive nitrogen balance. So as I've said here, pregnancy is certainly one. You're retaining more protein in the form of the fetus to meet the needs of the fetus than you are excreting. Bodybuilding. So if you're putting on lean muscle mass, then you're in a state of positive nitrogen balance. If you're growing, so if you're undergoing a growth spurt, if you're a child, you want to be in positive nitrogen balance. And if you're healing, so if you're wound healing or healing a bone or anything like that, you've just had a surgery, whatever, you want to be in positive nitrogen balance. Now, this doesn't mean you need to go out and eat so much more protein per se, but I think in various different situations, having a little bit more than what is required to put yourself in positive nitrogen balance would be prudent to do. So assessing nitrogen balance then. So how do we do that? Well, I did mention before that the assessment of nitrogen balance to assess nitrogen in versus nitrogen out is how the protein requirements were derived. So the recommended dietary allowances were derived. And this was done through a series of really intricate research studies in which they fed subjects, as you can see here, they fed many individuals known protein levels for various times. So over 3 to 14 consecutive days, they fed people known protein levels, and they collected all of their nitrogen that they excreted. So they know how much they're getting in their body, and they know what they excreted. Now, for most of the nitrogen balance studies, they really only do urine, feces, and maybe sweat. So they are missing a couple different things. But the idea is that for an individual person, you do these studies several times at different levels of protein, okay? And then you are able to figure out which level of protein provides that person with their nitrogen balance. And so these types of studies were done on many different people over the years, and they were able to figure out from a population level what the EAR was, so the average requirement of protein for healthy individuals, let's say. And then from that average requirement, they added 2 standard deviations, and there you get your recommended dietary allowance. So the recommendation is 2 standard deviations above the actual requirement. And the requirement is what is calculated through these nitrogen balance studies, okay, by measuring the known amount of protein in against the amount of protein that is out over up to a 2-week time period within people. Okay? So hopefully that's clear. If not, send me an email and I'll try and explain it again to you. Okay, so just to mention this here, we can measure nitrogen excretion in various ways, particularly from the urine. That's the most common way of doing it. And it is excreted as urea, creatinine, ammonia, uric acid. So again, anything that has a nitrogen in it, when we excrete it, it's likely from protein somewhere in the body, because that's really the only nutrient that has nitrogen in it. Now, there are several problems with these nitrogen balance studies. Primarily the fact that they're very difficult to do. So they're hard to collect all the nitrogen, as I mentioned. So, we're missing the nitrogen from the hair, we're missing nitrogen from nails. In females, we're missing nitrogen from menstruation. So there's various other ways in which, or other measurements that we could do that we're missing with some of these nitrogen balance studies, and they didn't necessarily collect them all. So they're difficult to do. Keeping someone in a metabolic ward for 14 days, providing them with all food that they require, and then repeating this several times within the same person is quite costly, okay? Trust me, as someone who does nutrition studies, it's really costly. Compliance issues, you don't know if people are compliant, whether they're consuming everything they need to consume, all of the above. So, and the compliance issues are really, let's say you don't keep them in the lab for all 14 days, you send them off with food and with a jug to collect their urine and feces and stuff. You don't know how compliant they're going to be, so sometimes there's issues there. And it's for these reasons in particular that these nitrogen balance studies have been criticized in several ways, because they do tend to underestimate the output of nitrogen. And so because they underestimate the output of nitrogen for various ways, the main way being that they don't measure all of the ways in which nitrogen could be lost, like I mentioned, this actually leads to an underestimation of the requirement. Okay, so this has been discussed as the main reason why people actually think that the RDA, or the recommended dietary allowance, is low, because of the underestimation of the total requirement for nitrogen, because nitrogen excretion is underestimated. So as you can see here, there are some issues with assessing nitrogen balance, but nonetheless, it's still quite an intricate scientific process, and it's really, really interesting. If you want to read more about how these are done, there's several studies that have been published about this, including the actual DRI book on how protein requirements are derived. You can take a look at that as well. And I think this is the last slide for today's class, but I just wanted to introduce you to this newer technique of measuring protein balance within the body. So this is another way of assessing the recommended dietary allowance, or the estimated average requirement, the EAR for protein. Okay, and so this newer technique is called the IAAO, or the Indicator Amino Acid Oxidation Method. And I've written down here exactly how it works. But essentially, they feed individuals, so it's a similar type of process to the nitrogen balance studies. You bring individuals in the lab, you feed them for several days or however long a particular amount of protein. And then in this case, you're actually measuring their breath that they give off. And what you've done is that you've labeled, you've given these individuals an isotopically labeled essential amino acid. So you know that it's one that your body doesn't make. Okay, and these are stable isotopes, not radioactive isotopes or anything, but these are stable isotope labeled essential amino acids. So in this case, they like to use phenylalanine, which is one of the aromatic essential amino acids. And so, you eat a certain amount of the amino acid, and they measure how much of the labeled amino acid comes out in your breath. Okay, so remember that when you undergo respiration and as your metabolism is happening, one of the byproducts of metabolism is carbon dioxide. So the idea is that you can measure the isotopically labeled amino acid in your breath to get an indication of how much protein your body is using. So when your protein intake is low, the labeled amino acid will be oxidized. And this is detected in the breath. So this is to mean that it is not incorporated into your body protein. So when your levels of I'll say this again, when your intake of amino acids are low, you're not providing your body with enough amino acids to make body proteins. And so at this point, the labeled amino acid is being shunted off to be oxidized instead, and it's not being incorporated into body proteins. So that is why you see it in your breath. So when your intake is low, you see it in your breath. Then as you increase your intake of proteins, this is protein grams across the bottom. Again, you can see it in this graph here, protein intake increasing across the bottom. As you increase your protein intake, you end up seeing less and less of it in your breath as carbon dioxide. And this is because more of it is being used by the body and incorporated into making other proteins. So as I said here, as protein intake increases, labeled amino acids will be incorporated into new proteins and less will be oxidized. So that is, less will be detected in your breath until you reach a particular constant level with which a stable level of the indicator amino acid is in your breath. And this is, this break point in this graph is considered the protein requirement for that particular person. Okay, and these are these are statistically derived. So you can see here the break point. These are some actual data from active young males that are 23, and you can see each individual person is represented by circles or squares or whatever, depending on the amount of protein they were given at the time. So what you end up with is just a bunch of circles and squares and triangles, filled in circles, whatever, all over the actual graph. And then statistically, yes, you're able to derive this break point in the graph. So you can kind of see here there's like a U-shaped situation happening, or a backwards J rather, in which case you can assess the break point and look down at the x-axis and figure out what the protein requirement is for these particular individuals. So you can, again, look at it within one person, or you can plot a whole bunch of people on the same graph and assess what you would get. Again, these are active young males, and what they found was the break point was 1.2 grams per kilo of body weight per day, and the upper 95% confidence interval, which for the most part corresponds to the RDA, was about 1.4. So this is quite a fair bit higher than what the actual RDA is, which is 0.8. So that's why they're saying in some cases the RDA is underestimated, and that this new method of assessing protein requirements, the IAAO method, actually yields much higher protein requirements. So something interesting to think about. I think we'll be seeing more from this method soon, and perhaps even a change in the RDA, particularly for older individuals. All right, so with that said, thank you so much for listening, and I look forward to seeing you again for class number 4.